right, so looking forward, so this Sunday, we've got Monday's point as usual. Unit 2, we've wrapped up almost all of Unit 2. We just have a couple of minor things to add now, so I think most could probably finish the Unit 2 concept check already. And so the concept check for Unit 2 is also due this Sunday. The mini project for Unit 2 is due next Sunday. So now all the mini projects from this point on are going to be a little more open-ended. So the idea is just to look through the algorithms that we have discussed in that unit. So in this case, Unit 2, that will be so immunocomputing, which we talked about today. So adaptive immune system, or sorry, artificial immune systems. And we talked about starting in Unit 2, CMA, ES, so the evolution strategies, and then we had genetic programming, So if so that now we're at the point where several of the mini projects are dropped. So I'm basically asking you, I think it's like to do 2 or 3 mini projects between now and the end of the semester. But it's up to you which ones you do. You can do more, but it just, I'm not asking for you to do every one of them, but for each one, and it kind of explains this in it, you basically find an algorithm we've talked about during that unit, and then find a way to demonstrate something interesting about that. So for this multi-modal optimization algorithm, it preserves diversity. I'm going to compare it to a normal genetic algorithm, and you see it all collapses to one point, but when I turn on the multimodal algorithm, it finds all the optimized. So we set up a little demo. So the goal for mini project 1 was to sort of give you an example of like I sort of set the demo up for you. So I tried to sort of say, how do we demonstrate how recombination works, how mutation works, and the general framing of a GA. And so for each one of these, you could say, well, could I come up with a little demo that I could, in 1 or 2 pages, be able to demonstrate some interesting feature of that algorithm, either just interesting on its own or maybe compared to some benchmark. So if you need help with any of those things, you say, I really am interested in this topic that we covered in this unit, but I'm having a hard time, so I'd like an opportunity to implement this algorithm, but I was having trouble coming up with an idea, and feel free to reach out, I can help you with that. But this is just supposed to be a way to balance that, in this class we could cover a lot fewer things and then force everyone to implement all of them, or we could try to cover more things and then give people who are interested in certain topics an opportunity to practice with those. And I've gone with the kind of the latter. Then otherwise, looking forward into Unit 3. Unit 3, I've split it up into 2 concept checks. And so just to get them sequentially on your calendar, I've made the first one due this upcoming Wednesday and the next one due next Sunday. But both of them are effectively due next Sunday, because I set the availability for both of them to through Sunday night, so through that Monday morning. So this is just meant to sort of help you sequence it, so you see it on your calendar, is that this 3A comes up next week and 3B comes up next weekend. But if you want to do them both next weekend, then that's totally fine with no penalty. Canvas might mark it red as late, but that late doesn't do anything. So as long as it's in by the availability date, it's fine. So these are effectively due next Sunday, but just to get it on the calendar to sort of, for those who are trying to go with the pacing that I suggest, then the suggested pacing would do 3A by Wednesday and 3B by next Sunday. And depending on, I doubt we're actually going to get through all the things that I've put in the notes here for today. So if things kind of bleed over into next week, I might bump these a little bit, but this is the plan. Any questions about that, about mini projects moving forward? Anything else? Yep.
Should the mini projects try to explore something that is beyond what we covered in lectures, or usually it's including a concept in that?
For the mini projects, it's exploring. You're free to sort of try something novel, but the kind of what we're going to be looking for is how well have you demonstrated a concept which we've covered at a high level in the lectures. For the projects, though, the team projects, which be those 2-person teams that'll be due at the end of the term, those are the things where I'm asking you to find a nature-inspired algorithm that we didn't cover in class. So go to the literature, say, here's something that, I could imagine that if I was writing a syllabus for this class, I could slot it in somewhere and it would be an interesting thing to cover, and then tell me about that algorithm. So, we don't cover honeybee-inspired optimization algorithms. So maybe you want to do a honeybee or an artificial bee colony algorithm or something like that, or a chaotic catfish PSO or something like that. So these are things that exist. And so those are things that you can then come together, or you want to take a genetic algorithm and bolt on something that is significant to it, that is a lot different than we've talked about, and apply it to some application that, like, a lot of people use the projects at the end of the semester. They might be working on a thesis project that involves using one of the AI zoo platforms to do evolution of robotics in simulated worlds. And so maybe they want to use some of the algorithms that we've talked about in class to work on an alternative gate strategy for their simulated bipedal robot or something like that. And so you can say, well, the algorithms we talked about in class didn't quite fit my problem perfectly, but I found another nature-inspired algorithm that I think might work. And now that I have seen a bunch of nature-inspired algorithms, I kind of get how they work. So I'm going to try it and apply it to that project. So that's the idea. We extend in the big project, and the mini project is your opportunity to demonstrate your comprehension of the stuff within the lectures. Any other questions? Yep.
So for the final project, do we get a list of project from where the kind of topics from where we can select, or a list of papers? Because some of us, we don't have much exploration towards bio AI or something.
If you go, and I'll talk more about this as we get close to spring break, but if you already go, there's a final project module, and there's a page linked within it where there's a bunch of suggested algorithms where you don't have to choose from, but for exactly this case, to say these are nature-inspired algorithms that I know about, that I think have kind of some interesting twists and variations from the things we covered in class. And if you chose any of these, they'd be totally worthwhile to choose. But you can also go to Google Scholar and say, I noticed an interesting pattern that I saw in way starlings were moving around. I wonder if anybody's done anything with that. And you could go and say, search for, optimization, starlings, algorithm, or something like that in Google Scholar, and you might find something pop up, and you can explore that too. Anything else? Okay, so we left off talking about immunocomputing. So this is a broader category than what we cover in this little unit. So we're focusing on artificial immune systems, but there are a lot of people that are just interested in, not only from an engineering perspective, but from a computational perspective on biology. How is information processing done in these systems that have evolved in a wide range of taxa, from plants to fungi to animals, that are robust to attack noise, errors, and intrusions. So these are the things that detect when something's wrong and respond to it, and then possibly remember a particularly successful mitigation strategy in the past that can be then called up very quickly in the future. So how does that occur? And so the natural applications here, you see these in cybersecurity and anomaly detection. Like I said, there's Dr. Stephanie Forrest here in Sky, who runs a center in biodesign that's kind of focused on biocomputing, society, and security. She is an expert in these areas, very well known for her work in these areas, uses a lot of immunocomputing and genetic programming. And so we focus on artificial immune systems, or AIS. These are set to mimic, in biology, what's known as the adaptive or acquired or specific, these are all synonyms, immune system. So also AIS. So I mentioned that when we talk about immune systems in biology, there's two major categories. There's the innate immune system. So this is something that the mechanism, so if you look at the metabolic pathways, if you look at chemicals that are involved, genes that are involved, these are things that you can trace all the way back to very, very ancient in the sort of the tree of life. So you can find the exact same components in your innate immune system, in your body, in a fungi, in plants, in other vertebrates, and other invertebrates. So very ancient, highly conserved. The innate immune system is a general response to an invasion, to an intruder, to an error. It's what gives you a fever. It's what causes an inflammation. If you get cut, you get cut, you see redness, you see puffiness, your body is recruiting a homeostatic process to tend to that intrusion. And it seals it up. It seals it up temporarily, quickly, and then gradually it seals it up more permanently, and then the inflammation goes away, it's not red and puffy anymore, and everything's resolved. That is one component of the innate immune system. It will interact with the acquired immune system that we're about to uncover here, in that the adaptive immune system can actually tag things for Gesundheit, the innate immune system, to kill and move out as well. So it's sort of a general purpose security force that when it detects something's off, then it can cause these general responses that have been successful in maintaining the integrity of the organism. There's a lot of interesting things there. There's also still a lot of research questions that are not known about the innate immune system. It's a little bit like trying to study a cruise control system of a car when you've only seen it hold one velocity. So I would like to know, is this a PID controller? Like what exactly, how is this car holding its velocity? Well, if you've only seen it drive at a constant velocity, you know it is doing it, but you don't know how it's doing it. You have to perturb it to see how it then comes back. And so that means it's much harder to study these very homeostatic regulatory processes, because most of the time you don't actually see how they're doing their job, you just see that they have been doing their job and it's holding things steady. That's really what the innate immune system is doing. The one that we focus a lot on here is this adaptive or acquired or specific immune system. So this is a specific learned response to a particular infection. So this is when you get sick with the cold and your body responds, your body might, or if you get sick with the flu, your body might be sick, but then you won't necessarily get reinfected for a certain amount of time because your body now remembers what that flu is, and then it knows how to respond early before it becomes a problem. But it had to learn that this thing that entered your body that looked kind of weird, but wasn't weird enough to trigger too much of an alarm initially, that weird thing actually should have triggered an alarm. And in the future, it will trigger an alarm earlier, and it'll prevent it from growing and spreading. So that's the idea of the specific immune system. And we focus on, so we're vertebrates, and there's took two major lineages of vertebrates. There's kind of the jawless vertebrates, which are like hagfish and eels and things like that. And then there's everything else. And both of them have an adaptive immune system. Invertebrates don't have something we recognize as an adaptive immune system, but they have a much more complex innate immune system that has some adaptive features. So, but in the vertebrates, we have these adaptive immune systems, and the one that I'm focusing on is the one in our lineage of vertebrates, all the jawed vertebrates. And so, like I said, the similar functions exist. But most of the mimicry you see is of the immune system that is in you and me. It's in all the other jawed vertebrates that are out there. So this immune system provides immunological memory. So this is what I mean, that you get sick once, and then you remember for a certain amount of time that if you get reinfected, you hopefully won't get sick after being reinfected. Because what vaccines are built on. It's highly specific. So there might be a little bit of, like, if you get sick with one thing, if you then get infected with something that's kind of similar to it, you might get a good response, but generally the response is very targeted. And so you've got a lot of these things memorized over your lifetime, and the memory may live through your entire lifetime, or it may decay. And that memory, like I said, can be lost over time. And then there are these really interesting cases. So measles is in the news. I'm not bringing it up because it's in the news. It's always in the notes here. But measles is this really weird case where once you develop an immunity to measles, in most cases, it erases the rest of your immunological memory of all the other things that you had a memory of. So even though you might come out of surviving measles, then you can get a lot of secondary infections that you used to have immunity to, because measles does a weird thing to your immune system where it manages to somehow wipe out the memory of other things. So it's that secondary risk that makes measles particularly troubling. And so this is called immune system amnesia. So again, memory in there too. So understanding the computational properties here is very interesting. We're not going to be focused, so we're focused on the generation of variation and then how that variation is maintained in an adaptive way. And that's going to look very much like the genetic algorithms we've seen already. Now, in evolutionary history, generational history, we generate variations by mutation. And mutation is often mistakes in how things are copied, and those have a certain mutation rate, which can be leveraged by over long histories to generate new adaptations that can then be selected for and spread through a population. We need something more rapid than that and more controlled as well. And so so this is the thing that jawed vertebrates have that the jawless vertebrates don't. We have something called VDJ recombination. Now, I'm not going to quiz you on what, so I just wrote these here so you know they stand for something, variable diversity joining, VDJ recombination. Again, I'm not going to go through the details. If you take an immunology course, you can learn about, what V, D, and J are and how these mechanisms work. What I want you to know is that this is nature's pseudo-random number generator. So when nature creates new, I'll call them B cells in a second here, new antibodies, in fact, maybe I'll just take that off here, it has to create these antibodies that are variable. And these variations allow it to match things it's never seen before. And so it just needs to have this hypermutation. So that's what we call this, somatic hypermutation. So extremely rapidly mutating, but only this particular cell type. When these cells are created, they go through a process of recombination. Now, normally, we only see recombination when sex cells, when sperm and egg are created. That's when you get a recombination where you get half the genes from mom and dad, and they're shuffled for which half, which portions of those get passed on. This is so-called somatic hypermutation or recombination. So this is the one time where that recombination process goes on in normal cells, so-called somatic cells, non-sex cells. And so when you're creating these immune cells, these antibody cells, these B cells, these take your DNA, and there's a section of your DNA that's highly variable, and there is a mechanism that will take it and it'll open it up, and it will reverse it, it will chop it up, it'll reassort it, and it basically just creates all sorts of new combinations of your own DNA and then reassembles them together, and then that gets expressed as a phenotype at the level of these antibodies, the levels of these B cells. And so you get a high diversity in phenotype. And the trouble is you have to figure out which one of these to keep and which one of these to discard. And you need to maintain diversity because you don't know which ones you'll need in the future. What their goal is antibodies are looking for so-called antigens. Antigens are a structure on the surface of things that invade your body that shouldn't be there. So they are things that do not look like any cell that is normally in your body. So as we'll talk about here in a second, every cell of your body has a little shibboleth. It has a little passcode that I'm not going to quiz you on these things, but it has to do with this thing called the major histocompatibility complex. And so every one of your cells has a barcode, which is tailored to you. And we've talked about in sexual recombination, and there's, like we mentioned, that there's a sexual compatibility. And so in engineering design optimization, you may not want to cross two parents that are very, very different. You don't want to cross a flying vehicle with a ground vehicle, because then you get half a ground vehicle and half a flying vehicle, and it's really low fitness. Well, you also don't want to necessarily cross things that are really close together, because then it's sort of wasted recombination. And in biology, if things are really close together, then there can be other artifacts that occur that are very bad. And one of those is that if the barcode of your mom was just like the barcode of your dad, then after recombination, the barcode of the offspring would also look like them. So you would start getting a lineage of humans whose cells all look like that lineage. And that would mean that if you found a virus that could mimic that barcode, it spread like wildfire through that lineage, because that lineage would be unable to produce antibodies that could fight that virus, because the virus would always look just like those individuals. So one of the things that seems to be driving, like they've had experiments on college campuses where they have like a bunch of men wear white t-shirts all night, and they sleep in those white t-shirts and they take them off, and then in the morning they put a pile of these things up and they ask a bunch of women to smell the shirts. And then they'll ask the women to say, how stinky is the shirt? And across all the women, they all sort of agree that these ones stink more than those. But then they'll ask, do you get a feeling of attraction to any of these shirts? And in those cases, you find that there will be some women will be attracted or list high some shirts over others. And there'll be variation there. And when they look at the major histocompatibility complex of the women versus the men who wore the shirts, the women who rank the shirts higher have the most difference in their major histocompatibility complex as the men. And so the thought is that part of what goes on in sort of what appears to be arbitrary sexual attraction between two animals is actually looking for another individual where the offspring is likely to have a different a different barcode than both of the parents. Because that recombination will create a unique identifier that if they had two similar major histocompatibility complexes, then you could get all of this lineage, like having cells that just look like each other, which should just be a niche for a pathogen to come in and exploit. So there's all this really interesting kind of cryptography and security and password protection that is going on inside your immune system. And that's sort of what we're just brushing the surface of in this unit. All right, so I want to talk about some specific examples, two algorithms real quick, and that's all we'll cover in this unit. Then we're going to transition to multi-objective. But any questions about this background so far? Okay, so this first algorithm, this will be a simpler one. This is one that Stephanie Forrest, Dr. Forrest has done a lot with. This is called a negative selection algorithm. And it is based on the process of central tolerance, which I've already kind of hinted at. So the key problem here is we want to reduce immunological false positives. So like I said, you want to have you want to have antibodies floating around in your bloodstream, ready to match invaders, but you don't want them to ever accidentally match your own cell type. If that happens, you get what's called an autoimmune disorder. I mean, that's why, for example, that's why Michael Jackson had very light-colored skin, because his immune system started to attack his pigment cells. And so once that occurred, once his immune system had accidentally gotten triggered in that his own melanin-producing cells were invaders, then over his lifetime, he gradually lost the ability to produce the dark skin color. And there's lots of other autoimmune disorders that affect the nerve system, CIDP. There's a bunch of different things like that where it's just the case where your immune system is in hypergear, and it's to the point where it is now has a false positive. It has started to attack a part of your own body. If you get an organ transplant, the major histocompatibility complex of that organ will not match your own. That's why you have to take immune suppression agents, because it will constantly get attacked because it doesn't look like it's supposed to be there because it doesn't know the password. So you have to tell your immune system to stop checking for passwords. And the problem with that then is that you're now prone for infection because it's letting everybody in, it's not checking for passwords. So a big problem here is how do we build these antibodies and have them have variation that can spot things that we don't know we need to spot. Like they've got variability out there that could match things we've never seen before, but never match ourselves. And so in the immune system, the body creates randomly varying antibodies through this VDJ combination. So they look for different cell structures, and what they look for varies from cell to cell to cell through this VDJ combination. Before being released, so it's actually kind of when they're created, they go to your thymus, and in the thymus, there are a bunch of test examples of self. So there are a bunch of cases, known good cases, which should never be matched by an antibody. And it will test all of your antibodies against these. And if any of those new antibodies match these known examples of self, they're basically set fitness equal to 0. They're removed, they're reabsorbed, they're discarded, they're not allowed to roam free throughout the rest of your body. So this is what we call central tolerance. It's filtering out self from randomness. VDJ is giving you a white noise of a bunch of random things, but you know yourself is matches in there and some of those random things. So then you have a comb of examples that you sort of sweep out any of the possibilities where this random process accidentally matches your own histocompatibility complex. So that's central tolerance. So then negative selection, it's based on this, the negative selection algorithm, this is for threat and anomaly detection. So you have a process that generates random classifier strings. So what do I mean by that? You can implement these lots of different ways. These could be genetic programs that take an input, so this input could be a sound file that you've recorded from a factory. It could be a trace of activity from a network, a TCP dump, from a network or something. So it could be it could be an image from a camera. And so these classifiers, they can be implemented in lots of different ways. They could be a neural network, like I said, or a genetic program, but you're built a program that basically just says yes or no, I've matched or not. And you generate, and it has randomness in what it matches to. And then you assign 0 fitness to any that match a set of self strings. So if you have a bunch of inputs that should never ever trigger these things, you test them all against that. And if any of those test cases come up true, so that they match, then you discard that one and you don't allow it to be used. Otherwise, you keep all of the rest and you use them to identify inputs that warrant extra investigation. So I mentioned this example. I think I'm going to have you read this example in one of the upcoming concept checks, where an earlier paper of Dr. Forrest is they were monitoring a drill press acoustically. And so that is an industrial drill press. The drill's being used all the time automatically, and they would like to detect when it's starting to go awry, when it's starting to operate outside of normal parameters. And so they recorded a bunch of drill presses that were within parameters, like they're fresh, they're new, all the experts say they're good, and recorded diversity of those as known good parameters. And then they just kept those in a bank. And then they generated a bunch of random classifiers, and just as many as they could, to hold in memory, and then tested each one of those classifiers against the known good drill press sounds. And if any of them matched, they got rid of them. And then those classifiers could just be running continuously. And if they start noticing something that matches one of those classifiers, they know that it's not from the known good set. And those random classifiers, they're just looking for random noise. So it shouldn't match anything that it hears. But if it starts matching something that it hears, then we know that something weird is going on. Now, of course, it can't be totally random. So it's more like you get like a random perturbation off of the normal case. And so you're going to effectively throwing away the ones that are very close to normal, that match acoustic patterns you actually hear, and then keeping those, you can use a generative model to do this, and keeping those weird ones that are weird enough away that if you happen to hear anything like that, that even matches a little bit, then it's worth having someone to go and check out that drill. Again, you don't have to do this in manufacturing systems. You can also do this in cybersecurity. You notice that there's somebody contacting this machine a lot more than normal. Like you can do this any situational awareness application where you have a sample of normal activity that a classifier can run on. And if you generate random classifiers and they start triggering, then you know that something might be off. And then you can run this process continuously. So theoretically, just like your body is constantly generating random antibodies, you can also generate new random classifiers. Keep checking them against the known good pool and just keep doing this. And then that way you might gain the ability to learn new threats later as you generate more randomness. So you don't have to do this all in one shot. So that's negative selection. Questions about that? Mimicking the central tolerance system. Yeah.
So we are only making like a whitelist system that allow these guys in, but anyone else that's not in the whitelist, keep them separate for further check. That's all we need.
Whether we call this a whitelist or a blacklist, it's almost like we're blacklisting classifiers that look too much like us. So in some ways we are whitelisting stuff that looks like us, and then the ideal would be that we only whitelist things that look like us. But the problem is the complement of things that look like us is too big. So we have to instead generate a bunch of examples of things that we can blacklist. And we're hoping that we generate enough examples of things that we can blacklist that there's a pretty good chance that if we get any actual intrusion, it will be in our blacklist. Does that make sense? Other questions? All right, let me reshare. Sorry about that. Thought it was about to go to sleep, and then I accidentally put it to sleep. All right, negative selection algorithms. Was there a question? Yeah.
Do you have more examples about how to generate the random classifier strings? Because you mentioned neural networks, and I can understand you have some sort of you give it some info and you train it to output one if it's known good, but then if you're not providing any adversarial examples, then it might just learn to classify everything as one.
Yeah, so the idea here is that you wouldn't necessarily train the neural network. You would just generate random weights for a neural network. And those random weights, so let's say you've got we will get to, for those who have not been steeped in the many layers, so to speak, of neural networks, we have a major section on brain and spine and stuff. We'll talk about neural networks. But those who've seen behind this curtain a little bit, one of the simplest neural networks would just be a single layer of weights. And you take a dot product, a similarity of those weights with an input. So if you have an image that has 128 pixels, you could create 128 weights, and that 128 weights would be basically, if you had an image you wanted to classify, one of the things you could do was put that image, those pixels, the picture of the cat, encoded in your weights. And then if you take those weights and you multiply them with the pixels of an image, if they match, then you'll get a very high score. But if they don't match, you'll get a very low score. So in that particular case, you could say, well, I could just generate random images, so random strings of weights. And if I generate enough of them, then some of them may match certain patterns that actually exist in nature. So that's kind of the thing, is that I don't have, if I was doing a normal training of a neural network, I would have a bunch of known input outputs, like these inputs should give me a 1, these outputs should give me a 0. I do the training, and then I shape the weights to best provide those outputs for those inputs. In this case, that's kind of being done by the negative selection itself. And it's doing it in a very different way, in that I'm creating a population of random neural networks. And then of that population of random ones, I'm just seeing, are there any neural networks that have accidentally matched something that, that might be something I don't want to match? And then everything else I just assume is, if that ever occurs, that would be a bad thing. And it's just like this anomaly. So there's a lot of junk. There's a lot of waste in this process. And so maybe you have a smarter way to generate these random classifiers, these random neural networks. Maybe you use some diffusion process or something like that. There's things that you could do here to try to have a more intelligent kind of set that kind of adds prior information. And then this is sort of, it's kind of like, this is a little Bayesian. Like if you do, like, we kind of assume uniform priors. Like we have no idea what good or bad looks like at first, but then we filter that uniform prior through a bunch of known good cases, and then we get rid of those. So our posterior is like uniform minus good. But if you did happen to know that if we're going to get an attack, it's probably going to look a little bit more like this normal behavior than that normal behavior, then you could perturb, you could build a really good classifier for the normal behavior and then randomly perturb the weights of that classifier around it. That makes sense. Okay. So yeah, a lot, just like the genetic algorithms, we're depending a lot on randomness and populations. You have to think in terms of the ensemble. If you think in terms of each individual one, you think that's crazy. A random neural network is useless. And it is. A single random neural network is useless. But if you've got an ensemble of them, and they cover enough of a possible outcome space, then there's that tiny chance that your one random neural network actually does something useful starts adding up, so that your ensemble has an appreciable chance of actually spotting something. Okay, so that's negative selection. The other big category of artificial immune systems, where I'm actually going to give you some pseudocode here, is clonal selection. And so sometimes you can follow this after negative selection. So you've found a bunch of antibodies that seem to be you've got now a suite of antibodies, and now you actually start learning that there are some antigens out there that are bad. So this was like, I have known goods and I want to reduce my false positive rate. Then you can say, well, now I want to actually reduce my false negative rate. So if I start building a library of known bads, then how do I reshape my antibodies so that they're best suited to catch all of those known bads? And that's clonal selection. It's a more positive selection here. And this will look a lot more like a genetic algorithm. So the idea here is that we're going to start out with a set of S antigens. So these are anomalous patterns we want to match. Someone's already told us that these are things that, these are attack patterns. Like, we may not know how to match them, but we do know that they exist, that these enemies exist, and we have some way of putting a test case of this enemy into our system and then seeing if we can detect it. So we've got our white hat hackers or something like that. That's our kind of our S set of antigens. And we're trying to build a set M of so-called memory B cells. And these things, if they're done very well, they will match any one of these antigens we throw at it. There will be a B cell that matches it very well. Some B cells won't match, but there'll be at least one that matches it very well. That's our goal. Okay, so in this algorithm I'm about to show you here, this is based off one that's in the published literature called clonal, so like clonal algorithm. But I think a lot of things that follow clonal selection, it's a similar pattern. So the idea here is then the first thing we do is we generate a random set of B cells. So these aren't our M cell. This is a random internal latent set that we're going to filter through and gradually select good candidates to add to our M cells. So these are like elites, these M, and then the B are sort of like the individuals we're just trying out. So we generate random ones. And then for every antigen in our test set that this is a bad actor that we want to match against, we are going to calculate the fitness, or in clonal, they refer to it as affinity, of all B cells to that antigen. So this is like taking the dot product, if you want to think really simply about it, of the antigen with the B cell. But think about it, remember, the B cell could be a complex decision tree, and the antigen could be some dynamic input that runs into that decision tree and generates an output. So it's not necessarily as simple as like a dot product. But the B cell is a process that can be mutated that generates some alarm state that we can call affinity. So it'll say, how well do you match this antigen? And it might not match at all, it might be a 0, or it could scale up to a 1 or something like that, or anything in between. So that's kind of like fitness. So once we have that, we're going to do something that is akin to selection in a genetic algorithm. We're going to take, there will be a hyperparameter you set, just like our parents. So you can say, select the highest affinity B cells for this antigen. And we do that with proportional cloning. So proportional cloning is just like proportional selection, but it's proportional to affinity, and we don't do any crossover. So we take all of these, so a high affinity B cell is going to produce lots of clones, and a low affinity B cell will produce very few clones. That's what I mean by proportional cloning. You can think of it as just like roulette wheel selection based on affinity. Yeah.
What is affinity, the effectiveness of that solution?
You can think of it as effective. Affinity is just a degree of match. So high affinity means that you've built an antibody that is really good at finding this antigen. And a low affinity means you've built an antibody that for this antigen doesn't see it at all. So then you do proportional cloning, and you place all your clones into a new set C. And then for every clone in C, you mutate those clones at a rate inversely proportional to their affinity. So high affinity clones don't get hardly any mutation. They don't change much at all. Low affinity clones then get more mutation. So the things that seem to be a poor match for this antigen end up getting clones that are a bigger deviation from the B cell that they came from. So now we've got a bunch of these clones, and we are going to determine affinities of these new clones that we generated from mutation. And then from there, we're going to take all the clones that we generated, we'll copy them into B, our population of this latent population of antibody candidates. Then we have to figure out which one of those we're going to store in M, which is our actual memory B cells that we stick around and keep around. And so we're going to copy the highest affinity clones into M, our elite group that maintains there. And then we take the lowest affinity B cells and we replace them with more randomly generated alternatives. And then we loop. So that's clonal right there, is that you've got this sandbox of B cells that, and then these temporary kind of playing sandboxes on C here, where you generate all these clones and you put them back in the sandbox. Each one of these sets usually is limited in size. And so you're not growing these things infinitely, but you're choosing who's a member of them. And so you're going to get lots of variation in B, but whenever you hit on really good ones, you store them in M. And then the hope is that over time, M is going to have members that are good matches for many, many antigens. Although B will constantly kind of chase its own tail. So the B group is going to be like, it's going to be shaped for the first antigen in the set. and then it'll go the next loop through this for loop. It'll then get reshaped for the next antigen, reshaped for the next antigen. So this B is this cloud of antibodies trying to follow all these antigens. And as it discovers good ones, they get siphoned off and then put into M. So that M gets this diverse set of antibodies. That's clonal. Questions about that?
Are these memory B cells like, is it generally so are you trying to optimize for like all of the antigens simultaneously?
That's right. Yeah, in fact, I'll make a comment about that on the next slide. But you can think of this as, this is getting much closer. The reason I put this here, our next unit is on multi-objective optimization. We have a unit after that on multi-modal optimization. And this is getting to that. This is showing we have algorithms where you're trying to do multiple things simultaneously.
Like vaccine or immunizations.
It sounds like, well, you said immunizations?
Vaccine and how immunizations work.
Well, that would be sort of, it's a little like presenting a bunch of antigens and then waiting for your body to do this. Yeah.
Yeah, so basically, the main goal is to get the M sets of cells, like cells, and the rest are just randomly get into back to the main algorithm. And then for each run, for each antigen, it's again like I just want to know the gist of this whole algorithm is like for each antigen, it runs through and generate M cells, the elite cells, and then the rest go back to the algorithm and gets the rest of the antigens.
Yeah, so there is an assumption here that each antigen is a good warm start for the next antigen. So this random B cell, I could have taken this and put it inside the for loop and randomized it with every antigen. But the assumption here is that these antigens are within the ballpark of each other. They're all coding for similar sorts of threats. So it's actually maybe better to, once you've learned a set of B cells that work for one antigen, that they might actually be better off than a totally random set for the next antigen.
That means we can say that it goes back to the population, just picks M cells, then go back to the
Right, copies those M cells off, and then they get here, and then the rest go on. And in those who had low affinity to the last antigen, they might have high affinity to the next antigen. Questions about that clonal or clonal selection? Yep.
They're like I guess like the number S and M are these just like hyperparameters or?
Yeah, the size of S and M, so these are sets, but the size of these sets are hyperparameters. And so, well, S is an input, it's just a list of antigens, but the size of M, how much space you have in your immunological memory. So, like, I don't know a lot of the mechanistic details. I think people are still figuring out how measles erase your immunological memory. But one thing you could think that measles might be doing is it forces, it causes your body's version of this to somehow empty out these because there's a finite immunological memory. And then you gain a lot of ability to respond to measles, but it's at the cost of emptying out this finite memory there. That's totally hand wavy, and I'm not an immunologist. I have no idea if that's actually what's going on, but that's a conceptual one.
There are ways antigens like some part organs to respond to.
Yes, but that's actually getting into the next unit. So, but that's not what's we do in clonal. But clonal, you can view it as inspiration for some of the algorithms that we're going to be talking about, where you can do exactly that. Yeah.
So you're studying B cells, like it's just a randomly named group of cells, or it's something like a biological term for this B cell?
Well, yeah. So in biology, they're called B cells. Yeah, memory B cells. In fact, that's the term there. There's also killer T cells. There's sort of a taxonomy of these things. They have overlapping functions and specialization. It's a really fascinating system. It's not C though, that is invented. So C was invented, but the memory B cells, that's inspired. Other questions? All right, so putting that all together, in both negative and clonal selection, there's a big focus on maintaining diversity. And this is going to become really important for So we're going to eventually get out of the genetic stuff, but any other further mentions of genetic algorithms, the focus will really be on not using a population to do one thing, but using a population to do lots of things. And this diversity maintenance is going to be really important, avoiding collapsing on one solution like we've seen in the previous uses of these things here. And if I pull on that thread a little bit more, and some of the questions have already led this way, if we were to sort of imagine if clonal selection was an optimization algorithm, what would its fitness function be? And I would say it's maybe something like this, where I've got an affinity function for every antigen that I put in here. And ideally, I would like to just maximize the sum of, over the set, I would like to have a maximal affinity for all of these antigens. And so one way we can think of doing that is just adding up all of the affinities. So I want the sum total affinity to be as high as possible. That can be viewed as my fitness function. But then if you think about that, if there is a physical constraint that means that an antibody for one antigen is going to have limited ability to have high affinity for another antigen, then this sets things up so that we get these sort of fitness, these local maxima, which are sort of that don't, where each hill is sort of equally good. So the idea here is like if I have one antibody that perfectly matches one antigen, and then I have another antibody that perfectly matches another antigen, then both of those antibodies have the same fitness. Because if it's the case that one antibody can really only match one or two antigens at a time, then you have to choose which one or two antigens you want to match. But so long as you get one or two of them, then you're going to have the same amount of fitness. So when we think about a population-based optimizer, what we really want to think about is I would love to have a population where these antibodies are on one fitness peak, these antibodies are on another fitness peak. So it's almost like my fitness function is shaped like this, where each one of these peaks is the location of an antigen or an antibody that matches an antigen. And I would like to have, if I have a population of a lot of antibodies, I would like to have that antibody spread out over all of these peaks. And this right here is so-called multi-modal optimization. And so that's something that we'll talk about in Unit 4. Is when, if we've got population-based optimizers, what can we do to them so that they don't all collapse on one peak, that they just find all the peaks? And there are problems like this one, where finding all the peaks is maybe more important than finding one of them with high confidence. Now, what we're going to transition into now is multi-objective optimization, which would be a slightly different way to frame this, where instead of viewing this as a combined sum of affinities, I could say, well, I want to sort of maximize all the affinities simultaneously. I don't want to sum them together. I have no way to kind of weight them together. It's just always good to be higher in each one of these. And that, we'll see what the distinction is as we go into this. But that's my transition to the next unit. Question?
Do we just quantify the peaks, or do we want to set a threshold?
So you're saying, like, if you have this multimodal fitness function, maybe you could just set a satisficing threshold and say as long as you're above that?
Especially when if we're talking about like defense or anomaly detection.
Yeah, so we're going to talk actually about this idea in right here in Unit 3, and then actually the next couple of slides, if we'll get to today or next week, on this type of idea of what we're called satisficing. So, but basically you can rewrite a fitness function so that it, imagine that instead of saying this was affinity, it's affinity minus target is what you're trying to maximize. And then so this idea would be that as long as, there's ways to create optimization objectives that strongly penalize you if you're under target, but then above target basically flatten. So it creates sort of a fitness plateau that once you go above that target, then you're fine. And then that would be, if you're trying to encode that sort of good enough into an algorithm like this one. It would just be, how do you write the fitness function so that it has that nonlinear, so it's like a fitness function, but it kind of looks like, if this was your genotype, your fitness function might rise up and then it just flattens out and then falls. And then anybody who lands on this plateau will have equal fitness and be likely to be selected. So you have this issue of, you want to make sure you have a low drift algorithm, because then the drift can cause this diversity to go away. So you just get a random individual. But as long as you've got something that doesn't have drift, then if all of these have equal fitness, then they'll maintain representation. But we'll get to maybe better ways to do that with multi-objective. I mean, part of these questions about, like, we're going to see that we're going to be tempted to what we're going to call scalarize everything when we think about multi-objective, but that's not the right way to think about multi-objective. And once we introduce so-called Pareto optimization, it shows us a whole new world of thinking about multi-objective and thinking about satisficing constraints and so on. That's much more useful to us and to the stakeholders that we serve as engineers. All right, any other questions about these immunological examples? Yeah.
I have like confusion about multi-modal, multi-objective. Like, can you give an example of what is a multi-modal and what is a multi?
Yeah, so multimodal is single objective. So, but that single objective has multiple peaks. So, whereas multi-objective is someone's just come to you and said, if you ask somebody, you could ask somebody what type of car do they like, and it turns out that there's 8 different types of cars that they would like equally as well. And some of them are good at this, some of them are good at that. But whatever the combination of traits, whatever you ask their personal affinity for those cars, then they'd be like, well, if it has this suite, I really like it. But if it has this totally different suite, I like it just as much. That's kind of like the single objective. I'm trying to make that one person happy. And there's multiple ways to make that one person happy. What multi-objective is more like is if you have multiple people, for one thing. And so you want to say, like, well, this person, like, I'm shopping for a car for the family, and the one member of the family has a certain set of preferences, and another member of the family has another set of preferences. And so in that case, it's actually impossible to make both of them happy simultaneously. So how do I make a choice in that case? That's multi-objective.
Like in multi-objective, you have to make choice, and even if it's like finalize it will for the single, like as you said, like out of multiple objective, only single thing should be selected, or a kind of combined.
Well, ultimately, when we're talking any decision problem, a decision problem is fundamentally a problem about making a choice. Mm-hmm. And in multimodal, that means that there are multiple choices that are reasonably good. And in multimodal, you might just want to find all of the choices that are reasonably good. Whereas in multi-objective, there are multiple things that the system views as important, but there is no obvious one way to say this thing is the best, because we have one ranking scale, so we can't sort things. It's impossible to sort things in multi-objective because we have no way to weight these things together. Here we're saying that all the affinities are equally important. But imagine if we drop that. Like, there's this question about weightings. Here we're saying they're equally weighted. What if we don't know the weightings? All we know is that you want high affinity in all of them. And there might be a weighting where one of them is a common cold and the other one is like fatal. And so maybe the fatal one you really want higher affinity in, but you just don't know that ahead of time. So how do we deal with that where we don't know the weightings? Yeah.
Can we assume like multi-objective to be a compound objective function where it's a linear combination of all the existing?
That's what I call scalarization. And also we're about to get to that. And that is the way you initially think of it, but that's what we're talking about. I'm going to try to liberate you from that thinking. So multi-objective, that is single objective thinking to multi-objective. So like when people start doing that, it actually isn't multi-objective anymore. Because how do you create that combination? That adds a level of subjectivity. Like, again, I arbitrarily added these up with equal weights. But you could say, well, where'd the equal weights come from? Like, well, it's just easier for me to write on the slide. And so it exposes my subjectivity when I sum things together. So we need a way to create numerical optimizers where you do not add that subjectivity in. How does that even work? That's what we're going to be talking about next.
So can we define some heuristics to find the weights for the different objectives that we are combining?
Well, that's the thing. There will be no weights. At the end of the next unit, when we talk about Pareto optimization, it will be a way to optimize multi-objective problems without even having to imply that weights exist. So we'll have to see how that works. And the hint is it's necessarily set-based. Single objective is really being driven by a point-based optimization approach where you're trying to find the best. In order to find the best, you need to infer that there's some weights that exist. In multi-objective, we get rid of the point assumption and say, no, what our real problem is excluding objectively bad choices. So that all we're left with is a set of choices that is objectively good, but leaves enough room for a human decision maker to add their subjectivity afterwards. So multimodal or scalarization, linear combinations, there you're adding subjectivity in to the problem definition. In Pareto optimization, which is the big part of Unit 3, we are factoring subjectivity out and putting it at the end of the process so that we can have a conversation with stakeholders about how to do the weighting after we've excluded things which no one should choose. So we'll see how that works. But any other questions about immunology? A lot of these other multimodal, multi-objective we're about to get to. All right, so that's a great transition to thinking now about multi-objective optimization and multi-objective evolutionary algorithms. And so a little background here. This is going to require us getting a little bit of a background in mathematical decision theory. So mathematical decision theory is a mathematical framework for describing how rational agents should select among alternatives, make decisions. I say should, this is a so-called normative framework. It is not descriptive. There are, a lot of you might have heard about behavioral economics and things like that. Those are frameworks which borrow a little bit of this, but those describe how real decision makers do make decisions. And, under the neurological constraints, psychological constraints, and so on. This is what decision makers should do if they were truly rational, if they were unconstrained. And so the challenges here that I've highlighted the ones that we are going to focus on, but if this is a class that was all about mathematical decision theory, we would do the non-highlighted things as well. And so these all together, uncertainty is a big part of it. So we're going to say if you have got multiple players and you care about all of their objectives together, and they need to make decisions which are aware of the other individuals that are playing, they have uncertainty about what those individuals are going to do, what the value of those things are for the other individuals, and so on. So that's an important part of this that we're not going to be talking about, but I just want to mention is there. Competing objectives will be a major focus, and strategic interaction. We're not going to focus on how agents could work together to form collaborations, but that is something that fits within this theory. We're going to talk about utility functions and their relation to payoff functions, objective spaces, and equilibrium concepts. So I haven't defined what an objective space is. That will come up when we start talking about Pareto frontiers. And then the major subfields that we're going to focus on are game theory and multi-objective optimization. So MOO, multi-objective optimization, is a subfield of MCDM or MCDA, multi-criteria decision analysis. So we're focusing on a small set of MCDA and a small set of game theory in this unit. Those are the two big things that we talk about in this unit. Okay, so let's get to it. So let's start with a little game theory. This is in part to give you a background, an important tool for modeling things that occur in natural systems and how we use computation to think about these spaces, but then also how we use this game theory to help us in some numerical problems that actually start looking like games. So we'll talk about in a second here, generative adversarial networks. So these are when you pit two neural networks against each other, and those two things end up playing a game that is well described by game theory. And the equilibrium concepts we have here and the solvers for these equilibrium concepts end up being better at optimizing these networks than, good old-fashioned gradient descent, for example. So there's a bunch of tie-ins here. So just bear with me for a while that this is not just me, talking about something that I think is interesting just because I think it's interesting, that it will be relevant. So the idea here behind this game theory, we have the players, or if you like to consider them agents, that's fine too, and they have so-called utilities. So I'm going to use this function that I'll sometimes say utility, I'll sometimes say payoff. In this class, we're considering risk-neutral individuals. So these are individuals that don't care about whether they've got a lot in the bank or a little in the bank. They sort of make the same decisions. So in this case, utility is the same as payoff. In general, payoff is what somebody hands you. Utility is how much you value it. So if you're risk neutral, if somebody gives you $5, you value it the same regardless of your current state. And so that's why I'll just use those interchangeably. But in general, they're not interchangeable. Utility is how much you value the payoff. And so these utility functions, this is the value of, so each one of these players can make an action, which we quantify with these variables. So player one can make action X1, player two makes action X2, and so on, all the way up to player N. Now, the utility, the payoff that player one feels is a function of its own action and everyone else's action, but it only has control over its own action. So that's what we're showing here. This is a function of player one's action and everybody else's action together. So that's where we get player one with utility U1, same with 2 and N. Okay, so if you have a game like this, then we have to sort of say, well, what would happen in this game? And is there a notion of an equilibrium in this game? In other words, is there a notion of a set of strategies that if everybody played that strategy, that the game would sort of stop? That people would stop changing their strategies. And that's the so-called Nash equilibrium. And so the mathematical description of this is for every player, I, and every alternate strategy that player could play, Xi, we're describing the Nash equilibrium. So I guess I should have said this up front. The Nash equilibrium is a vector X star. And that is all of the strategies that all the players are playing at the Nash is X star. So Xi star is what player I is playing at the Nash. And Xi without a star is an alternate strategy that the player could be playing instead. So for every player and every alternate strategy that they could play off the Nash, then the utility to that player, and this is a common notation used in game theory, where rather than writing all these arguments over and over again, I focus on the one argument I really care about, and then if I do a minus in the subscript, it's all of the other arguments. So rather than me saying x1, x2, x3, comma, da, 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 comma, xi, comma, da, 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 comma, xn, I just do this instead. So I'm saying I've got xi at the Nash, and then everybody else at the Nash. So this is saying that for any alternate strategy that player I could play, its payoff at the Nash is at least as good or is better than the payoff it would get by playing the alternate strategy if no one else played an alternate strategy. So this is what we refer to as a unilateral change. So no unilateral change can benefit the player making that change. So this is a kind of a no regret condition here. So if the player is playing that, they might imagine a world where other players would play other things. And it's like, gee, if I could just get that player and that player and that player to play these different things, then if I played this different thing, I could be doing better. But if you can't move anybody else, if those are stuck, they're all playing the Nash, then I don't have any regrets because I can't control them. So if I'm stepping away from my strategy, I'm going to be worse off. So I'm okay. Although I can imagine a way I could be better off, I don't have any control of getting there. So the things that I'm under control of, then I'm doing as best as I can over those things. So that's what I mean by you cannot benefit from a unilateral change. And you can have, you can view this as multiple competing objectives. And so game theory is very closely related to multi-objective optimization, and the notion of Pareto optimality, which again will be one of the core things we take out of this unit, applies both in game theory and in multiple optimization or multi-objective optimization. So you can view these as players, or you can view these as multiple objectives in a complex problem you have. All right, any questions about that? Of the definition of a Nash? Yeah.
Could you just repeat what the star above the Nash equilibrium means again?
Well, so this is the notation I will use to indicate that this is a Nash equilibrium. So if it has a star, that means it's a Nash.
And then in the inequality.
So this over here, this means that every other player, so that's the minus I, is playing their component of the Nash. So X star is a vector that has, so if I write it up here, I could say that X star internally is X star 1, X star I, all the way up to X star N for our N players. And so all I'm saying here is that I is playing an alternative strategy, so it gets rid of this, replaces it. But everybody else plays what they be playing at the Nash. So if we were playing a game and you had to choose a number, and so I chose 6, you chose 2, you chose 4, and so on. So 6, 2, 4, and whatever. And if we decided that was a Nash, then what that would mean is that if any one of us, like let's just say me, if you stick with 2 and 4 and so on, if I decide to change from 6 to something else, but you stay at 2 and 4, then my payoff will go down, or it won't go up. And then, but likewise, we can do the same game with you. If I stay with 6, you can decide to perturb off of 2. And then so if you go to 1 or 3, then your payoff won't increase, it might go down. So that's the Nash. So for all of us, it happens simultaneously. That's what the Nash is. Questions about that? Yep.
Can you explain again why is it greater than the RHS?
So the utility, we're saying here that the Nash utility, that's this one, is better than the alternative. So there is no alternative that can increase you more than the Nash. That's why the condition for the Nash is a greater than. Okay.
There can be multiple equilibriums.
Yes, there can be multiple Nash in any game. There can be no so-called pure Nashes in some games, but there can be so-called mixed Nashes, and that's kind of getting outside of the scope of what we were talking about in this class. But there definitely can be multiple Nashes. So multiple ways. And then, the question is, how do you get from one Nash to another? Is that possible to do? Some Nashes can be better for the group, but if you're stuck in the Nash that's worse for the group, there may not be a path to get there because it's an equilibrium. All right, other questions about that? So where we're going to pick up from next time is we're going to then apply these concepts and then bring in Pareto for a discrete game and then for a continuous game. In the continuous game, we'll see gradients come back, we'll see so-called variational inequalities, and we'll see things that apply to modern machine learning. And then that will allow us to then transition to true multi-objective optimization, where we finally define this idea of an objective space, and we stop thinking in terms of strategies and more think in terms of trade-offs. So that's where we're going. But we'll stop here for now. And have a good weekend. And I think still, yeah, unit 2 stuff is due this weekend. And I'll see about whether the unit 3 stuff, whether it'll shift a couple of days. I just have to kind of look at what's in those assignments and how far. So if you have any questions, feel free to come up. Otherwise, like I said, have a good weekend.